Okay, next in our multi-factor ANCOVA unit, we cover blocking and covariates. These are also just additional factors, like additional treatments, but they are experimental design factors. They are not treatments that I vary purposely, but they are additional factors that I add to my analysis of variance to increase the signal-to-noise ratio. So it allows me to account for some unwanted variation in my experiments. And the idea of both is the same, just in one case the covariates are actually numeric variables and the blocks are categorical variables. Blocks can be spatial, temporal, or have another individual designation that we'll talk about in this video. So blocking and covariates are actually very commonly used. They are very useful design features, so this video gets a star for a new important concept. You probably always in real-world experiments either implement a blocking structure of some kind, or you want to measure some covariates in any kind of sampling design that you could potentially later use in your analysis. Okay, so covariates are variables that I'm not interested in, but that nevertheless have an effect on my response variable. I can't control them entirely in my experiment, but I don't really care about them, so this is not the subject of my research. So just to give you an example, if I have a variety trial, like the example that we used before, that can be laid out in a field with a completely randomized design, but I may have some topography here. So I may have humps and I have little valleys and maybe nutrients and water gather down there, and at the top of those little humps the soil is not as good, and that may influence my individual treatments here, right? So I can go out and measure this. Let's say we are going to measure soil moisture in regular intervals, and I can get a covariate here that also influences my response variable, the yield. So perhaps when it's wetter and there may be slightly more nutrients, those particular plots have higher yields. But if I measure those, I can account for this additional variation. So covariates can be measured before, during, and after the experiment. So if you find that you have some strange results and you go back out and see, oh, this is what's going on here, you can still measure those and then add them to your analysis after the fact. This you can do in experimental situations, but this also applies to sampling designs. So for example, what I drawn up here is a number of lakes with different sizes, and I'm comparing control lakes with no disturbance and perhaps some forestry or agricultural operation nearby that may pollute the lake to some degree. I want to know what the effect on my species richness or species diversity is. So I'm measuring the species diversity here, but lake size might also have an effect on species diversity. So I could use the lake size as a covariate in my data table to account for this additional factor that I don't necessarily have control over in my sampling design. So I may not be able to pick lakes that are all equal size. So covariates can be taken under all kinds of circumstances. So especially sampling designs that are logistically very intensive, it pays usually to measure all kinds of covariates just in case they play a role. So if you suspect that something else might influence your response variables, where you take your samples, so take stock of the environment, survey the environments where you collect your data. So the implementation in R is really super simple. It's just like any multi-factor analysis of variance. You simply add your covariate to your model and that's it. R will do all the hard work for you. The one thing you have to be conscious about is the variable type. So if you have a numerical variable, R will do an analysis of covariance. And if you have a class variable, R will do an analysis of variance with it. So having your class variables defined as actually a factor variable and double checking that your covariates are a numerical variable never hurts. So just run a structure command of your data table before you run your ANCOVAs so that there are no surprises. And that's also one of the reasons why you don't want to number your varieties or number your lake exposure. So always start those treatment levels, so those class variable values, with a letter, then you don't run into problems that the linear model does a covariance analysis. Good, and last but not least, go with a plus here, not with a star. The covariates are not necessarily variables that you're interested in the first place, so it's probably not a good idea to waste your degrees of freedoms on interactions with your covariates, but it can be done. So you could test for interaction effects. But they should make sense, right? So maybe in the first example, they would make sense that variety and soil moisture do have some interaction effects, so that some varieties respond differently to soil moisture than others. But in the second example, clearly that would be a case where you wouldn't want to test for interaction, because what would be the biological mechanism to create inconsistencies here in response? So these are likely additive effects. Okay, let's talk about blocks. Blocks are essentially exactly the same idea, except that I make the covariate part of my design. So this would be a completely randomized design that we talked about, where there's no particular structure. But what I can do is I can arrange my treatments into blocks. So each block has all treatments within it, randomized. But then the blocks are laid out across putative environmental gradients. So for example, if I have a slope where I suspect maybe, again at the bottom, I have higher water availability, and that may influence my response variable, then I would lay those blocks out perpendicular to my slope. So that's called a randomized complete block design, and it's a very common design. As soon as you have any kind of perceived environmental gradients, and you almost always do, lay it out as a randomized complete block. And if you think about it, it's really a big brain move to do it this way, because you get the benefits of a covariate, except you don't have to measure anything, right? So this is all automatically taken care of, instead of going to every plot and measuring soil moisture and nutrient content. The blocks capture that just as well. So one important thing to remember, that people don't necessarily realize that that's the case, but blocks don't need to be contiguous blocks like I've drawn here. They should just have similar environmental conditions. So if I have a river system here, I may want one block here with available areas that are close to the river, then another block 2 over here, a little bit further away. Again, a block that's laid out here, close to a river, and a block further away from the river. So they don't need to be connected at all. It's good to have them not spaced out too far, because that also may drive differences that you don't see. Maybe soil types change over here relative to here. So don't space your blocks out too far, but you should try to lay them out in such a way that they capture the environments. So the shape of the blocks, whether they are squares or rectangles or triangles, is completely irrelevant. The only thing that matters is that you place your treatments into groups that have similar environmental conditions. And the general idea of blocks can actually be extended to other things rather than spatial blocks. So anything that can influence your measurements can be accounted for by blocks. So for example, if you have a big experiment and you can't finish measuring it in a day, or if you know that your measurements are sensitive to the time of day where you take them, let's say it's a physiological measurement on photosynthesis, so it matters whether you measure it in the morning or at noon or later. So what you do in this case, you take your measurements within the blocks that you already have, or if you don't have blocks, you can just create post hoc blocks. So you start with 3 treatments that you measure in the morning, then 3 treatments that you measure at noon, 3 treatments that you measure in the afternoon, and then the last set of replications you measure the next day. So those would be temporal blocks, and what that does is that effect would be removed from your noise term, so it would be accounted for in your analysis as a block effect. So that reduces the variance that's left in your error term and increases your signal-to-noise ratio and your statistical power. Now, if you already have a block design, so that's one reason why you might want to lay out things in a block design, you can use this block structure to assign other factors that may vary. So for example, if you have a number of assistants who help you measure this, then assign a block to each assistant or multiple blocks to each assistant. So if they have slightly different takes on how they measure things, or if there's some subjectivity involved in your measurements, for example, if it's an estimation of vegetation cover or something like that, then any kind of difference among how your observers take those measurements will be accounted for by those blocks. But you don't have to use those blocks. For example, if you have, let's say, a complicated instrument and you go through your plantation and maybe here it breaks and you have to call it a day and go home, fix your instrument or buy a new one and come back another day to do the rest, then you can simply have a post hoc block that you put in. So one block represents all the measurements that you've taken with your first instrument, and all other measurements are assigned to the second block where you use the second instrument. So those things are possible even if you don't have a block design to start with. And blocking would also apply to sampling designs. We use this lake example where you have control lakes and lakes with close-by agricultural operations, but they're also placed in different regions or different ecosystems that you suspect might have an influence on your species diversity. So in that case, you can call a region or an ecosystem a block in your experiment and account for some of the variants that are not actually driven by the treatments. Now, one important thing you have to keep in mind if you do blocking in a sampling situation like this one is that those blocks are likely unbalanced and incomplete. And that's perfectly okay. So R can handle this, especially the mixed model procedures that we'll mention in a minute, have no problem with this kind of arrangement. The only thing that you really have to watch out for is that you have at least one comparison within each block. As a block 4, the way I've drawn it here, that would not work because it misses a second treatment. So it doesn't matter if it's unbalanced, so if I have 4 controls and 1 agriculture treatment here, but just having 2 controls like in block 4 here, that's not going to work. So in that case, you would have to pool these 2 into 1 block or remove this entire ecological region. So incomplete is fine. That may not be the best example, so let me think of another example. If you do a wildlife study and you compare 5 different habitat types and how that influences your species composition or how that influences an endangered species. So if you have 5 habitat types, it doesn't matter if they're incomplete. So you may have habitat 1, 3, and 4 here, 5, 1, and 4 here, 1, 2, and 3 here. That's all perfectly fine. In analysis, that's not an issue, but you need at least 1 pair. That's important, otherwise that block would be worthless. So again, implementation in R is really straightforward. So what you do is you just add your block to the factors in your multi-factor ANCOVA. And again, I wouldn't specify my interactions. In this case, both the block and the main treatment would be class variables. And analysis of variance doesn't know the difference, so it just uses it as another treatment. So what happens is that the variance that's due to that block effect gets removed from the error term, right? So your error term gets substantially reduced if you have a large block effect, and that increases the statistical power for your treatment effect that you're really after. Now there is another version that you can use here, and that's called a linear mixed model. The option that you have here is to define an effect as a random effect. So rather than a fixed effect that you fixed as an experimenter, this is just a random effect that is not under your control. And that means that you don't actually want to estimate the mean or test for significance. So there is no reason actually to waste degree of freedoms. So in mixed models, you can define this as a random term and it won't even show up in your analysis of variance table. That increases your statistical power even more. So by all means, if you work with a randomized complete block design, do use a mixed model. It's only a 2-letter difference. Oh no, there's a few more here. So this is how you specify a random effect. So putting it in brackets and putting the 1 dash in front. So we're not going to get into the theory of this here in much detail, but those are safe to use. So just to let you know what those terms mean and when mixed models are applied. So a fixed effect, that would be your regular treatment. It's fixed by the experimenter or purposely chosen in a sampling design. Your random effect has potentially an influence on your response variable, but you're not generally interested in it. So we account for it with blocks, but we don't estimate block means or test for significance. And so you can have fixed effect linear models, so you're just putting treatments in there that you're actually investigating. You can have completely random effect models, so if you're just interested in variance components, you don't have any fixed effects. So that's very common in plant breeding or genetics, where you're simply asking, what's the variance component that's due to genetics and what's the variant component due to environmental effects. And mixed models have both. And the most typical example is a randomized complete block design. So you have a treatment and a block where you don't actually care whether your blocks are significantly different and by how much. So this is just an experimental design factor. Now, mixed models are actually a big deal. It's good to know how they work. So if you're an experimenter, you may want to get more deeply into this. But it's not for this particular reason. So you could analyze a randomized complete block design just as well with a plain linear model. There's virtually no difference. So all you basically do is save a few degrees of freedom by not estimating block means. So in practice, this makes no difference whatsoever. The reason why mixed models are important is the underlying algorithms that are used to estimate parameters, so that are used to estimate the means and estimate the linear model coefficients. So they're much more advanced than what the LM procedure in R can deliver. So it's worth getting into this if you have very large, complex designs that are unbalanced with tons of missing values. Then those mixed model procedures, they shine for completely different reasons. But for the purpose of normal designs, you might as well use the linear model. It'll always give you exactly the same answer as the mixed models would.